Hi everyone, welcome to another class on the topic digestion and absorption. Today we would be continuing with the structure of the alimentary canal, in which we have already studied about the buccal cavity and the pharynx. And in the buccal cavity we have studied about the structure of teeth and the tongue. Now today we will move on from the pharynx towards esophagus. So let's begin today's class, and in which we will be studying esophagus, stomach, small intestine and large intestine. So first part starting with esophagus. Now esophagus, as we can see here, that this part here is the buccal cavity, then that is the lower part of the pharynx, that was laryngopharynx, and from the inferior end of the laryngopharynx the food pipe begins, and that food pipe is esophagus. So it is almost 25 centimeter long and it is posterior to the trachea. So that means in the front there would be the windpipe or the trachea, and behind it there is the esophagus. And this esophagus, it passes all through the thoracic cavity, and there is a muscular diaphragm which is partitioning our thoracic cavity from the abdominal cavity. So this diaphragm is pierced through by this esophagus, and the region where it pierces, that region is called hiatus. And through that hiatus, the esophagus will pass into the abdominal cavity and open into the stomach. So basically, we can see that esophagus is extending from the end of the laryngopharynx and up till the upper end of the stomach, and that is the cardiac part of the stomach. Now, if we see the histology of this esophagus, so there are few differences in the histology. That is, the mucosa, that is the innermost layer, has upper 1/3 region where there is non-keratinized stratified squamous epithelium. While in the case of mucosa of the entire alimentary canal, we had mentioned that mucosa is having columnar and glandular epithelium. But here the upper 1/3 part is having non-keratinized stratified squamous epithelium. Then another difference here is that in the upper 1/3 part there are skeletal muscles, while the entire muscularis otherwise is made up of the smooth muscles. And then not only that, where the skeletal muscles are present, there the two plexus, that is the Auerbach's plexus and the Meissner's plexus that we have talked about, they would be absent. So the plexus are absent in this region. Then both ends of the esophagus, they have sphincters. And when we say sphincters, so sphincters are made from the muscularis only. And in the muscularis, we had mentioned that there are longitudinal and circular muscles. So at the region of the sphincter, the circular muscle would be more developed, so that when it contracts, the entire lumen will be narrowed down and that opening will close. So that kind of a structure is called sphincter. So we have to remember that sphincters are made from the circular muscles. So both ends of the esophagus have sphincters. The upper one, that can be simply called upper esophageal sphincter, and the lower one, that is called the lower esophageal sphincter or the cardiac sphincter, because it is leading the food bolus to pass into the cardiac stomach, or it is also called gastroesophageal sphincter because it is at the junction of stomach and esophagus. And then one more histological difference in the esophagus, that serosa is absent, and instead of serosa there is a connective tissue layer only, which is called tunica adventitia. So in this case, this serosa is replaced by tunica adventitia, while in case of serosa we had mentioned it is mesothelium plus connective tissue, but here it is only connective tissue. And as far as these sphincters are concerned, there is one kind of disorder which certain people may have, and that is if this lower esophageal sphincter is not competent. That means it doesn't relax when the food bolus is passing into the stomach, and such a condition is called achalasia cardia. So here we can mention this term achalasia cardia. And this achalasia cardia is a condition where the lower esophageal sphincter is incompetent. So what would happen? Food will keep on accumulating in the esophagus, and that can even cause the rupture of esophagus. And last but not the least, we can mention that esophagus, where the mucosa is concerned, it doesn't have any digestive glands, but since it is mucosa, so certainly mucus secreting glands are present, and mucus is secreted, which is a slimy substance constituted by glycoproteins, and that lubricates the passage. So there are no digestive enzyme secretion, but mucus secretion is a common factor. Now moving to next part of the alimentary canal, and that is the stomach. Stomach, also called gaster. Now this we can see the shape. It is a J-shaped organ and it has 4 regions. Now these 4 regions, we can mark them here. This upper region, this is the fundus. It is filled with gas. And the region just after the esophagus, that is the cardiac region, and it is so named because it is near to the heart. So just below the diaphragm, above the diaphragm, the heart is present, and this is just below the diaphragm. And then this major part, the larger part, this is called body. And the remaining part is called pylorus. Now this pylorus is having a broad region which is called pyloric antrum, and the narrower region, that is the pyloric canal. So in this way we can see these 4 regions, and as we said, these are fundus, cardiac or cardia, and this is body, and this region is pylorus. And here we have already mentioned that where the esophagus ends, there is a sphincter here, and that is the lower esophageal sphincter. And at the end of pylorus, and where the duodenum, that is the next part of the alimentary canal, starts, here again there is a sphincter that is called the pyloric sphincter or pyloric valve. Now, what are the basic differences in the histology of the stomach? If we see that, so here we can see that the wall of the stomach, where muscularis externa is present, in that wall there are, instead of 2 layers of muscles, there are 3 layers. As we can see, the outermost longitudinal, then middle one circular, and inner to the middle one there is a oblique muscle layer. While in rest of the alimentary canal we had seen there were only 2 sets of muscles, that is longitudinal, outer one, and circular, which was the inner one. But here there are 3, and that third additional layer is oblique muscle layer. And all 3 of them of course are smooth muscle layers. The inner side, that is the mucosa is showing these kind of folds here, and these folds are called rugae. And these rugae can be seen when the stomach is empty. Once it is filled with food, then the stomach lining stretches and then these rugae disappear. So they would be seen only in the empty stomach. Now coming to the mucosa of the stomach, that mucosa of the stomach is folded to form branch tubular glands. So these branch tubular glands are the gastric glands, and these glands have certain types of cells, the cells which are secreting certain constituents in the gastric juice, those will be the exocrine cells. And here the exocrine cells which are present are mucous neck cells which secrete mucus, parietal or auxentic cells which are producing HCl and Castle's intrinsic factor. Their functions we'll be just mentioning. The chief cells or the peptic cells or the zymogen cells, they are secreting the inactive precursors of proteases, that is pepsinogen and prorenin, and there is also small amount of gastric lipase, which is a lipid digesting enzyme. Now, the purpose of the mucus here, of course, is to protect the lining of the stomach from the HCl itself, because this mucus is alkaline. HCl, we will see that it activates pepsinogen and prorenin into active forms. Castle's intrinsic factor has importance in helping in the absorption of vitamin B12. Then gastric gland, they are also having endocrine cells. Some endocrine cells are there, in which one of them is the G cells, which secrete a hormone, gastrin hormone. Now this gastrin hormone stimulates these exocrine cells to produce their secretion. So that means the HCl secretion and the enzymatic secretion will be stimulated under the effect of gastrin. And there is another one, argentophin cells, or which are also called enterochromaffin cells, and they secrete serotonin or histamine. Serotonin is a vasoconstrictor and histamine is a vasodilator. Now, let us use this information and answer a question. The type of cells present in the gastric glands which secrete intrinsic factor. Now, intrinsic factor or Castle's intrinsic factor is secreted by the parietal or the auxentic cells. So, the answer here is 3. The peptic cells and chief cells are one and the same, and they secrete pepsinogen, prorenin and gastric lipase. So, here our correct answer is 3rd. Moving on to another question. What will happen if the secretion of parietal cells of gastric glands is blocked with an inhibitor? Now, parietal cells, as we know, that they would be secreting HCl and Castle's intrinsic factor, and if it is inhibited, what happens? Now, enterokinase will not be released from the duodenal mucosa, and so trypsinogen is not converted to trypsin. So, in this case, the secretion of the parietal cells is not affecting the secretion from the intestinal glands. So, that is unrelated. Gastric juice will be deficient in chymosin. Now, chymosin is just another name for renin. And the gastric enzymes are secreted from the peptic cells or the chief cells, not from the parietal cells. So, this is also not correct. Then another one, gastric juice will be deficient in pepsinogen. This is again ruled out because of the same thing, that peptic cells are not affected. Now coming to the 4th option, in the absence of HCl, inactive pepsinogen is not converted to active enzyme pepsin. Now this would be correct because the HCl being secreted from parietal cells is carrying out this step, that is converting inactive enzyme to active enzyme. So this is our correct answer, that is option 4. Now, after taking these few questions, we can move on to some more aspects of gastric juice, and then we'll take up the intestine. Now compiling up the constituents of gastric juice. So here we can mention gastric juice, which is acidic, and the pH can range from 1.8 to 3, and normally it is within 2. Now this gastric juice contains hydrochloric acid, The purpose of the hydrochloric acid is that, of course, it kills the various pathogens which come along with the food, and we'll see that most of the bacteria are not able to survive in the acidic medium, although there are exceptions, like there is a bacteria Helicobacter pylori which is able to survive even in acidic medium, and that causes damage to the gastric mucosa resulting in gastric ulcers. Then this HCl converts prozymes or the zymogens, inactive enzymes, into active enzymes, and it reduces ferric to ferrous. So whatever plant food we take, like spinach, that is rich in iron, but all that iron is in the form of ferric, and this HCl is reducing ferric to ferrous, and we are able to absorb only ferrous. So if this absorption doesn't occur, there would be anemia, because iron is important for the synthesis of hemoglobin in our RBCs, and in the absence of ferrous, there would be anemia. The other one, that is pepsinogen, which is inactive enzyme. Any name which is ending with ogen, that is inactive, precursor of the enzyme here, and it is converted to active pepsin by HCl. And the purpose of pepsin is that it is a protease, protein digesting enzyme. Then prorenin, which is converted to active renin again by HCl, and it hydrolyzes the milk protein, that is casein. And the secretion of this prorenin declines as we grow, because initially all mammalian babies are totally dependent on milk, but once they start taking solid food, the dependence on the milk declines, and the secretion of renin also declines along with it. Gastric lipase, which will be splitting some of the triglycerides, that is the lipids. Intrinsic factor, the Cassell's intrinsic factor, that aids in the absorption of vitamin B12, that is cyanocobalamin. Again, a very, very important vitamin for RBC synthesis, and in the absence of B12, there is an anemia cause that is called pernicious anemia. And mucus, which protects the stomach lining. Now, if this gastric juices in this HCl is secreted in higher amounts, like if the person is having indigestion or for a long time they have not eaten anything, and this HCl, it regurgitates into the esophagus. As a result, there would be a burning sensation in the esophagus, which is called heartburn or acidity. So basically, when we talk about acidity, so acidity is reflux of acid from stomach to esophagus. Now let's move to next part of the alimentary canal, and that is the small intestine. Now the small intestine is the longest part of our alimentary canal, and it is divided into three regions. Now just after the stomach, this C-shaped structure that we see here, this is the duodenum. And this is the shortest part of the small intestine, and as we see, it begins from the pyloric sphincter, and then it is leading to the next part, that is the jejunum. And this jejunum is the middle region, and this is the one which we can see here. And if we see the internal side of the jejunum, it appears quite red. That shows that it is highly vascularized. And then the longest part, that is the ileum. It is highly twisted. The word ileum itself means twisted. It is the longest and the narrowest region. Now, if we take the histology of the small intestine, what do we find is, it is almost same as that of the GI tract, that is our gastrointestinal tract, but there are certain differences. One is, of course, there is lot of lymphoid tissue that is present in the lamina propria, although this is all along the gastrointestinal tract, but in ileum we have aggregated patches which is called Peyer's patches. And this entire lymphoid tissue which is found in our alimentary canal, that is called GALT, that is gut associated lymphoid tissue. And of course, the purpose of this lymphoid tissue is that through food and water, what all different infective agents we are taking in, some of them can be controlled here itself by the lymphoid tissue. Now, another important point here is that if we see the inner side of our small intestine, what do we find is that the submucosa is folded into these circular folds. And since the submucosa is folded, so that increases the surface area, then over this submucosa there is mucosa, which is shown here with the yellow color. The mucosa also is highly folded. So that increases the surface area manifold. Now these circular folds are called plica circularis. Plica circularis, or they are also called valves of Kerckring. These finger-like folds, these are called villi. Now, if we see the structure of the villi, this is how it will appear. So, this entire finger-like fold, this is the one villus, and that this is how it. These finger-like folds. Now, if we see the structure of the villi, this is how it will appear. So, this entire finger-like fold, this is the one villus, and inside this villus what we can see is that there are these blood villi increases the surface area manifold. Now these circular folds are called plica circularis. Plica circularis, or they are also called valves of Kerckring. And the folds of the mucosa, which are these finger-like folds, these are called villi. Now, if we see the structure of the villi, this is how it will appear. So, this entire finger-like fold, this is the one villus, and inside this villus what we can see is that there are these blood capillaries present. So, here there is an arteriole which is branching out into capillaries, and then that is opening into a venule, and that is then passing out. And in between these capillaries we can see this green structure, that is actually a lymphatic capillary, and lymphatic capillary here is called lacteal. And in this villus, if we see these cells, so what kind of cells will these be? These are epithelial cells which are columnar and glandular. And this columnar cell, if one cell out of here, I take it out, then this cell has again finger-like folds, and these finger-like folds are the microvilli. And so these microvilli give the appearance of brush border. So that's why we say that the alimentary canal is lined by epithelium which is brush bordered epithelium. So that is a columnar epithelium having microvilli, and that again is increasing the total surface area. So here we have a assertion reason type question where the assertion is gastrectomy causes iron deficiency anemia. Now gastrectomy, gaster word we use for stomach, tomy means to cut and ec is to remove. So gastrectomy would mean that by surgery the stomach is cut and removed from the body, and this is going to cause iron deficiency anemia. This is the assertion given. So first thing that we have to judge is whether this assertion is correct. Now if you remember, in stomach just now we have mentioned that HCl is secreted, and that HCl is reducing ferric to ferrous, making it into absorbable form. And if ferrous is not present, then in that case there would be anemia because of deficiency of iron. So certainly this statement is correct. If we see the reason, hydrochloric acid secreted by the oxantic cells converts ferric into ferrous and iron is absorbed as ferrous. So that is absolutely the correct reason. So assertion is correct, reason is correct, and reason is the correct explanation of the assertion. So our option correct answer would be when both assertion and reason are true and reason is the correct explanation. So this will be our correct answer, that is 1. Now let's move on to intestinal glands. Let's take up the intestinal glands. Now, in the intestine, we find two types of glands, that is the Brunner's and the crypts of Leverkusen. The Brunner's gland, however, are present only in the duodenum region, so these can also be named duodenal glands. And there is one more special distinction about them, that they are present in the submucosa. Now, this is important because all the other gastrointestinal glands, they are present in the mucosa, but this one is present in the submucosa. And they secrete just thick alkaline mucus, nothing else. So they are not secreting any enzymes, just thick alkaline mucus. And the purpose, I think everybody can very well understand, because the acidic chyme which is coming from the stomach has to be quickly neutralized, and that is why abundant alkaline secretions are required. And that is the reason that duodenum is receiving this alkaline secretion, as well as the intestinal juice, which is also alkaline, and it is also getting pancreatic and bile juices, those are also alkaline. So the combined effect of all these alkaline juices is able to neutralize the acidity of the chyme. The other glands, that is the crypts of Leverkusen, these are of course in the mucosa, intestinal mucosa, and they are located within the region between two villi. So we had just seen the structure of villi, and between two villi there is a narrow region which is forming a simple tubular gland, and that one is the crypt. Now these glands, they have one particular type of cells called the Paneth cells, and these Paneth cells, they secrete lysozyme. Now lysozyme is an enzyme which digests the bacterial cell wall, that is peptidoglycan. So this one is helpful in again controlling the bacteria which have come into our body through contaminated food and water. Then other than this, these crypts are also secreting number of digestive enzymes. And these digestive enzymes, they are not released into the lumen of the small intestine, but they remain attached on the epithelial cells which are having the microvilli. So they remain attached there, and that is why such enzymes are called brush border enzymes. So these enzymes of the small intestine, they are brush border enzymes because they remain attached to the microvilli surface. And the important enzymes present in these are aminopeptidase, dipeptidase. As the name suggests, they would be acting on the partially digested proteins. Disaccharidases, that means which is acting on disaccharides, like maltose is a disaccharide, that will be acted over by maltase. So maltase is one of the disaccharidase. Nucleotidases and nucleosidases. Here nucleotidases will be acting on nucleotides, which could be a deoxynucleotide or a ribonucleotide, and then when that nucleotide is converted into a nucleoside by removal of the phosphate group, then that is called nucleoside, and nucleoside will be acted over by nucleosidases. Now, the secretions of these two glands collectively is called the intestinal juice or the succus entericus, and it is alkaline with a pH of 7.6 or it may range up to 7.8. Now, coming to some of the important hormones as well. Now, from the small intestine, there are a number of hormones also secreted, which are called the enteroendocrine hormones, and the cells which are secreting them, they can be named enteroendocrine cells. Now, some of these important hormones, first one starting with secretin. Secretin also was the first hormone to be discovered, and that opened a new world to the biology, and that was endocrinology. Now, this secretin stimulates the release of bicarbonate carbonates from both of these glands are the acidity. So when that opened a new world to the biology, and that was endocrinology. Now, this secretin stimulates the release of bicarbonates from the pancreas as well as from the liver, and both bicarbonates from both of these glands are again important in neutralizing the acidity. So when the acidic chyme reaches into the duodenum, there the cells get stimulated and secretin release will start, and it is also inhibiting gastric secretion and motility. Now you see, when the food is stored into the stomach, it is stored almost for 4 to 5 hours, and slowly from stomach the chyme is released into the duodenum. So when the stomach muscles they contract, so that will be the motility, and that will be pushing the food ultimately into the small intestine. So all that is going to be suppressed, and also gastric secretion, where one important secretion as we know is HCl. So that is the reason that secretin is also called the nature's antacid. That means it is preventing the secretion of HCl. Another important hormone is CCK-PZ, that is cholecystokinin pancreozymin, or these days it is simply written as CCK. Now this term chole is always related to gallbladder. So that's how it gets the name, because it causes the contraction of gallbladder. In the gallbladder, the bile juice is stored. So when the gallbladder is contracted, that bile juice is going to be released and it will pass into the duodenum. And it also stimulates the release of pancreatic enzymes. So that's why we get the name pancreozymin. So here we can see, and these two are very important questions as well. If we are given a question on release of bicarbonates from the pancreas, the option has to be secretin. And if it is regarding pancreatic enzymes, then the answer is CCK. Another important hormone is GIP, that is gastric inhibitory peptide, where this I can also be written as insulinotropic, because that will give one more function of this GIP. So as the name suggests, gastric inhibitory peptide, that means it inhibits the gastric motility and secretion. And the other name if we take, that is gastric insulinotropic peptide, that means it is acting on the pancreas and stimulating the release of insulin. So whenever we use the word trophic, that is to stimulate. So insulin secretion is stimulated by GIP. Now earlier an older name was used, that was enterogastron, and enterogastron was just meant for suppressing gastric motility and secretion. Then another hormone, duocrinin. This is, as the name suggests, duo, that is only acting on the duodenal gland, and the gland which is specific to duodenum is Brunner's gland. So it is stimulating the Brunner's gland to release the alkaline mucus. And another one, enterocrinin. Enterocrinin, it stimulates the crypts of livercune to release the enzymes which are present in the succus entericus. Now we can use this information and solve a few questions. So here we have a assertion reason type question. Now in this, if we read this assertion, gastric emptying is slowest after a fat laden meal containing large number of triglycerides. Now in this case, fat digestion is almost nil in the stomach. So if the meal is very high in fat content, it will remain longer in the stomach. And the next thing, of course, here is that fatty acids in the chyme stimulate the release of both CCK and GIP. So when this fat laden chyme is reaching to the duodenum, there is more release of CCK and GIP, which both of them are slowing the stomach emptying. So here the assertion is also correct, the reason is also correct, and the reason is explaining the assertion. So in this case, our answer is going to be 1. So here our correct choice is answer 1. Now let's take one more question. Which cells of the crypts of livercune secrete antibacterial lysozyme? Now argentophin cells, these are the enteroendocrine cells which we had seen in the stomach, and similar cells are present in the crypts of livercune also. The other one, that is zymogen cells, these are of course which are secreting inactive proteases, and this term preferably we use in case of stomach. Kupffer cells we will be studying in the liver, and here Paneth cells. And that would be our correct answer. Now let's move to the next part of the alimentary canal, that is the last part, and that is large intestine. Now moving on to the large intestine. The large intestine, that is the terminal part of our GIT, and it is extending from ileum, that means the last part of the small intestine where it ends, and from there to anus. And the opening of the ileum into the large intestine is guarded by a valve which is called ileocecal sphincter or valve, because sphincter is made from circular muscles, and since it is acting like a valve, that is allowing the partially digested food or undigested food to move only in one direction and not allowing it to return, so that is why its behavior is like that of a valve. The large intestine is structurally divided into 3 regions, and those are the cecum, colon and rectum. And in the large intestine, some of the water, electrolytes and small amounts of vitamin K, they get absorbed. So let us take on the structure of this large intestine. So here we see the large intestine, and this is the ileum here. And this ileum is opening into the first part, that is cecum. And at this opening there is this ileocecal valve. Now this cecum is a dilated region. In our case it is very small, but if we see in case of herbivores, there the cecum is quite large where fermentation of cellulose takes place. Now this cecum is giving out a small finger-like projection, an outgrowth which is called vermiform appendix. Now in our case, vermiform appendix, as far as digestion is concerned, it is a vestigial structure. But this vermiform appendix is also filled with lymphoid tissue. But sometimes what happens, that undigested part, some seeds which we might have taken along with the fruits we eat, that may drop into this vermiform appendix, and then it will rot there, causing immense swelling with pain, and that is appendicitis, and that has to be operated upon, and then the appendix has to be removed. Now this cecum is also harboring number of symbiotic microorganisms, especially bacteria. Number of bacteria are remaining there, and there are certain foods which we have not been able to digest. For us they are undigestible, but for bacteria they would be raw material. So bacteria would be fermenting them. Now then, taking up the next part of the small intestine, and that is the colon. Now colon is having 4 segments. Here when it is rising up, this is called ascending colon. Then it runs through the width of the body, so that is the transverse. Then it is coming down, that is descending, and then it takes almost a sigmoid shape. This is called the sigmoid colon. So these are the 4 parts, out of which the ascending colon is the shortest segment. And this sigmoid colon is opening into the rectum. Now the histology of the colon shows some very special features. We can just mention them, and these are Tenei coli. Now Tenei coli are bands of longitudinal muscles. Now we had seen in the muscularis, there are longitudinal muscles all along the entire circumference. But here these longitudinal muscles, they are present in the form of bands, and there will be 3 such bands, and those are called Tenei coli. And because of this, the entire wall of the colon is forming pouch-like structures which are called haustra. And even the serosa here has folds in which fat is accumulated, and those folds are called epiploic appendages. So these are 3 structures which are specific to colon. Now we can again see these structures in the diagram. So here, this one is the Tenei coli. Wall is thrown into the diagram. So here, this longitudinal band that you see, the entire has, and these finger-like folds are filled with fat, histological variations that we are able to see in the colon. Then in the colon also, bacteria that you have learned till date. So that means it is a resident of colon, and Escherichia coli. And these bacteria don't have any benefit or loss, but they are secreting vitamin B12 and K. So vitamin B12 and K are produced by these symbiotic bacteria. There are nearly 400 different species of